We don't need to do that. I think we're live.
Great.
Yeah, it's exciting.
Well, it's great to be here.
Yeah, I'm just gonna make sure that everything is streaming okay.
Okay, super. Yeah, that's something you got to do on your end. That's the magic that happens over there.
Yeah. Is it good? I'm waiting for is anything happening?
It's still loading.
Okay.
Waiting for the A-OK.
Yeah. Well, it says that we have like 140 viewers, so I'm assuming that it's streaming okay, but well, they might let you know in the chat.
Okay. I think it's because I'm on Wi-Fi.
Okay. Well, we're just gonna keep chatting. I think we're okay. We have like 85 people coming in. So as long as someone is helping to monitor the chat, I think we're okay.
It's coming along. I don't see Soon in the background.
You don't. Soon is.
Oh,
Oh,
look!
there.
Oh, yes.
Yeah, there is Soon Raccoon.
Oh, so nice to see.
he's in retirement, enjoying his time.
Yes, his new state of residence.
Okay, so we're gonna get started because we have some people tuning in, which is really exciting. I am super pumped that we can be joined here today with Dr. Andy Warren from the University of Florida Museum of Natural History for this very special day that I feel like it's a holiday, like a birthday, but more special, sort of.
It's Wahydra graslieae Day, in addition to being International Women's Day, which was just a coincidence. So happy International Women's Day.
This is the best International Women's Day I've ever had. This is one of the best days I've ever had, period. I came all out with my butterfly earrings.
Oh, you're decked out. Yep, very nice.
So Andy, why are we having this special announcement today? Can you fill in our viewers really quick?
Sure. Yesterday, a paper came out in the journal called Zootaxa, which named a new species of skipper butterfly after Emily Graslie in recognition of her tireless efforts to promote natural history collections. And that's really why we did this for you, because you spent so many years really stressing the importance of natural history collections. And most of your episodes touch on it either indirectly or directly. And it's like, we were sitting around being like, wait a minute, how come nobody has named a creature after Emily yet? This is like way overdue. So yeah, it was like I can't believe that we were the first people to do it, actually, but we're honored to be. So as someone who just kind of fell into the role of natural history museum fangirl and was able to make that into a profession, I don't think I ever would have anticipated I would get this sort of recognition ever. The fact that it's an insect is like super exciting to me, and the fact that it's to be honest, no offense, a pretty nondescript looking insect is also really special. Like you have it there, actually. You can show it. Yeah, we have it actually right here. So here's a drawer of holotype specimens.
Wow.
And a holotype is the specimen which the definition of a species is based on. So every species out there has a holotype that researchers can refer back to if they have any questions about the identity of what they're looking at. And so this little guy here, that's Wahydra graslieae. And you'll notice everything in this drawer has either red or blue or yellow label on it. So these are all type specimens. And we don't tend to label type specimens until the actual publication comes out. So today we're going to go ahead and put the holotype label on Wahydra graslieae. If you're ready, Emily.
It's like a little ceremony.
It is. It's like a little ceremony.
It's like the nerdiest ceremony.
It's the nerdiest ceremony. It's the holotype labeling party here. So yay. I've got the labels printed out. They're here in this little envelope. Let me go ahead and get them out.
Oh boy.
And so it was official yesterday when the paper was published, but now it's a done deal because the specimen is labeled. So
that's exciting.
There's two labels here. One we're going to put on the actual specimen itself.
Mm-hmm. It has been pinned.
It's labeled in. So this little vial has the bits in it. the genitalia. So when you describe a new species, you have to compare it to close relatives and really document what makes it different. You see that it's a really small skipper and there's not a whole lot of wing markings. So a lot of the characters that make this skipper different from other Wahydras, they're characters that you see in the genitalia. So inside this tiny little vial are the bug bits, and we keep them together with the specimen, and it also gets a holotype label.
Very important to pin the genitals of the insect and to keep them with the rest of the creature.
Yeah, you definitely want to keep them together. So
you don't want to pin that holotype without its diagnostic features. That's
right. That's the rest of the specimen. Yep. So that's the complete holotype.
There it is. And now anybody who wants to research Wahydra graslie can come to the University of Florida and check it out for themselves.
Indeed. Yep. And there's a lot more to be learned about this specimen. So in the original publication, we included the DNA barcode, which is just a little snippet of DNA, which is used as sort of a quick and dirty way to compare species to each other. But we intend to try to get a whole lot more DNA out of that extraction so that we can do like a genomic analysis and really see how it compares to other Wahydra species.
That's exciting.
We're in the process of getting genomic data for other Wahydra types. And so this one is like, we can't wait to add the data and see how it compares to the other species.
Yeah. So can you tell us a little bit about, how this specimen was, how you discovered it, where it was discovered, and then how you were able to determine that it was actually a unique species?
Sure. So the specimen was collected by a researcher called Dr. Harold Greene, who runs the Yanayacu Research Station in Kosanga, Ecuador. And he picked this up back in 2004 and had no idea what it was. He just thought it was a cool little skipper. And I study these things. I've been studying skippers for about 20 years now. So we have a collaboration. He sent the skipper to me. And immediately when I saw it, I said, this is something completely new. It's just like not something that's been seen before. But the problem was we had no idea what genus it really was supposed to go in, because it really looks very odd for a Wahydra. Most Wahydras, these are other Wahydras here.
Wow.
And so you see most of them are actually more colorful than graslieae. Most of them have a lot more orange. So that's the reason why initially we didn't really realize it was a Wahydra. And so the next step was to dissect the genitalia. And as soon as we did that, it was obviously Wahydra. All the structures are the same form. There's just some minor variations in details in some of the structures, but it conclusively showed that, hey, this is a Wahydra, and it's a weird Wahydra, and it's a Wahydra unlike any that anyone's seen before. So typical for Wahydra, it's at high elevations. Most of them are Andean, usually in sort of cloud forest habitats where the weather is pretty crummy most of the time. And that's why they're rare in collections, because they're not encountered, because they're in these sort of hostile habitats. And they're not like plants. I mean, butterflies like this, they really only fly when it's sunny and during nice weather. And if you're there and it's rainy, or if you're there in the clouds, you're just not going to see them. And so that's why they're not encountered very often in nature.
Wow.
So the specimen was collected in 2004, but it wasn't until 2016 when we actually dissected the genitalia. And it's actually really common for new species to sit around and wait for someone to study them in detail. And it was my Brazilian colleagues, Eduardo Carneiro and Diego Dolbaina. They were here in 2016, and we had set up sort of an assembly line to try to identify and describe the sort of backlog of unusual skippers that I had, and we just needed to figure out what they were. So this was one of them. And Eduardo, who's the senior author of the paper, he's actually described several other Wahydra species. So when he made the call that, oh my gosh, I mean, he literally looked up from the microscope and said, it's a Wahydra, and we all go, oh yeah, that makes sense. It's a Wahydra. And so from then it just like, oh wow, it clicked.
That's cool.
Yeah. So as far as what made us decide to name it after you, well, they weren't aware of the brain scoop yet, and so we were sitting around watching the brain scoop that night, and it just sort of became obvious that you were like waiting to have a species named after you. So that's when we decided to go ahead and do it.
I'm truly honored. It was really special when you sent me an email asking how I would feel about it. It was like, it's really nice, because I think especially when we as like a YouTube creator, we're always making content to help, shed a light on the sort of work that happens behind the scenes at natural history museums. And I kind of go into this knowing that I'm never going to be like a viral sensation, like the kind of work that we talk about and the stuff that we feature is kind of as a niche audience, one that is incredibly loyal and very enthusiastic. And I wouldn't trade our viewers for the world, but it was really kind of special to be like, you know what? I think I'm the first YouTuber to have a species named after them. And like, that is an accomplishment that I'm pretty proud of. And that I think like really is a testament to the actually like the feedback and the enthusiasm from our viewers, because I wouldn't still be making videos 5 years on about natural history museums and the nuances of taxonomy if it weren't for the fact that there's like a real hunger and interest from people all over the world to know more about this. And so, that we can kind of follow the process about a specimen being discovered in a museum drawer and then kind of leading it through this whole process of being named and then deposited. And now it's available for research. Is really cool. And I just also really appreciate that there's so much we don't know about this little organism, right? Like the important thing I don't think we've touched on yet is that the holotype is the only known record of this species. Like that is the only one.
It's the only known specimen. And normally new species aren't named from just a single unique specimen, because what if it's an aberration? What if it's not really what you think it is? But in this case, it's such a unique skipper that it can't be anything else. If it's anything but a new Wahydra, it would be like a new genus, but you wouldn't name a new genus really after one specimen. It just seems to be a pretty different Wahydra. So yeah, I mean, lots of questions like, what does the female look like? The characters of the female are going to tell us a lot about how this species fits into the genus Wahydra and how it's related to the rest of the Wahydras. How about the larval food plant? Well, my guess is it's a bamboo, only because one or two other Wahydra species have been reared off of bamboo. And when I say bamboo, it's the genus Chusquea, which is super like hyper diverse in the South American Andes. Every 1,500 feet or so in elevation, the species of Chusquea replace each other, and all the butterflies that feed on them also replace each other. And Chusquea happens to be really good butterfly food. So one species of Chusquea might host 30 or 40 butterfly species.
Wow.
And you go up 1,500 feet in elevation, and those 30 or 40 butterfly species all switch because it's a different food plant then. So it's extraordinarily diverse down there in the Andes. And if it's a bamboo feeder, that could also explain why it seems to be really kind of rare, because these bamboos are real spottily distributed and they're confined to certain elevational zones. So if we can figure out what bamboo it feeds on, if it does feed on a bamboo, that'll make it a lot easier to rediscover the thing. You just go to where the bamboo is, and you spend a lot of time, and probably the caterpillars could be found on the food plant.
You have no idea what the caterpillars of this species look like.
No clue.
That's that's such an exciting surprise, because you could be out and you could find a caterpillar and then just be like, I don't know who you're going to be. Like, what do you do in that instance? Do you then collect the caterpillar and then like sequence the DNA off of that, or do you rear it?
Well, ideally, you would rear it out. If you're there for enough time, in situ, you would want to rear it out. If you can't rear it out, you can go ahead and preserve the larva and get DNA sequence out of it. And assuming that species or something related, that there are available DNA sequences out there, then you could narrow down the identification that way.
That's so cool.
But ideally, you want to try to rear it out to the adult.
That's neat. I mean, who hasn't like captured a butterfly or caterpillar in a jar and grown it up? It's such a special experience.
I would like to think that nobody has missed out on that, but I'm afraid some people have not done that yet. But the same Harold Greene who collected this your skipper, he's also the same person that has reared Wahydra. He's the only person that's reared Wahydra. So the only bamboo records that we have for the genus are actually from him also.
Really?
Yeah.
So like, how diverse is this group of insects, and how many people study them worldwide?
Wahydra or skippers? Well, Wahydra, there's graslieae is the 15th known species.
What?
Yeah. And really only 4 or 5 people have ever dabbled in Wahydra taxonomy. Wahydra, by the way, it's the word itself, it's a meaningless anagram from based on the name Kenneth John Hayward. And Hayward was a skipper researcher based in Argentina through the 1940s, 50s, and 60s. And in one of his papers, he noted that the species that we now have in Wahydra, they used to be in a different genus at that time. He says, well, these probably deserve a different genus, but he didn't name one. It wasn't until 1991 when a skipper researcher here in Florida, associated with the Allen Museum of Entomology, went ahead and described the genus Wahydra. And at that time in 1991, there were 5 species that he moved into Wahydra, 4 new species that he named at the time. So in 1991, there were 9 species of Wahydra. Since then, 5 more have been named. And then yesterday with graslieae, that's up to 15 species of Wahydra. But there's probably quite a few more, because given that the Andes are huge and that every 1,500 feet in elevation, the species turnover, I wouldn't be surprised if there's twice as many Wahydra out there as we actually know. Only time will tell. Yeah. Only time will tell.
Andy, we've got some questions from viewers that are coming in, which I think is exciting. So if you guys have a question that isn't answered by like going through the press release, we have links to all that information in the video description. But we've got a couple here that I think are really interesting. The first one is, are there any other species named after awesome people in that box of holotypes? And I want to kind of go off that question too and ask, like, how does one get a species named for them? Like, who is being recognized? Like, what sort of people are they? All scientists? Are they YouTubers?
Well, you kind of there's sort of all of the above. More often when a new species is named after a person, it's usually a scientist involved in that field, but not always. A few to a butterfly name, and so somebody got to select whatever name they wanted for that new butterfly. There are cases where new butterflies or animals are named after famous people, and I guess this would qualify for one of those. As far as this particular drawer of holotypes, there's actually quite a few here named after people. So this is Dalla austini. There we go. That's Dalla austini. George Austin was the previous collection manager here, who I collaborated extensively with up until the time he passed away in 2009. And then this is our tiny Steinhauser-eye, named for Steve Steinhauser, the person who named the genus Wahydra back in 1991. So these are 2 cases where active researchers in the field get species named after them. But also it's common to name a new species after local inhabitants of the region where they're from. So this little thing here, that's Artanis maya, and it occurs in the Yucatan Peninsula where the Maya culture basically originated. This one is also named after Austin. Any more famous people? I don't think so in this drawer. A lot of these are named after the place where they're from. So very frequently, like Texana or Mexicana, if it's from one of those places. So those are common names as well.
Cool. That answered that question. That's pretty cool. And then someone, I think we kind of touched on this a little bit, but if you could go into a little bit more detail. Someone asked, what makes a species a new species? And could it just be a mutation or something like that? So like, how do you know for sure?
Right. Well, how you know for sure is if there's anything else that's like it. And you sort of have 2 semi-independent lines of evidence. You've got the morphology. And so that's what the wings look like. That's the legs and the head, the antenna, and of course the genitalia, which in this case, the genitalia are Wahydra-like, but unlike any other species of Wahydra. Then the other line of evidence you have is DNA. And so in the original description, we went ahead and included the DNA barcode, but at that time, there were no other Wahydra to compare it to. So there was really no basis for comparison. Now that we're sequencing the genomes of more Wahydras, we're going to be able to use that barcode and compare it to other Wahydras. And so far, I mean, there's no other Wahydra that's that close. So like I said, I mean, once we learn more about it, it's possible that it's its own genus. But I think the chances of it being just a mutation of something already known are like close to none.
Wow, that's really cool too. Is like there are no absolutes in science.
No, there really aren't.
Thing. Like
that's right.
This is named for me today, but maybe evidence surfaces later on, and it turns out that this is more or less related than another, and maybe the name changes. But at this moment in time, like this is just the reflection of what we know about this organism, and
right.
The change that happens in the future is another like signal of progress in science, which I find really exciting.
It totally is. And like one possible scenario, if say Wahydra graslieae is more widely distributed than we think, say it's from southern Colombia into Peru or Bolivia, there could actually be geographic variation within that range, and they could end up naming various subspecies of graslieae. that's the other thing that happens. Like when things get better known and we understand the geographic variation better, then subspecies can be named. So, you know, the sky's the limit. Yeah.
I feel like I'm the mother of like so many little baby caterpillars out there in the world right now. I'm so proud.
Yeah, well, you know, you are.
That's exciting. So someone had a question too. This butterfly was collected in 2004. What was I doing back then? What would I have said if someone told me in 2004 you'd have a species named after you? In 2004, I was in 8th grade. I was watching a lot of Sailor Moon and eating a lot of Pocky Sticks in Rapid City, South Dakota, generally being pretty angsty as a 14-year-old. So, and really like kind of jaded. I think if someone at that time had told me there'd be a new species named after me, my first reaction would have been like, haven't we found all the new species yet? Right. But how little did that girl know? But no, I mean, it's really, I could have never anticipated that there would be a time where I would have a species named for me. And it's just not a thing that, like, I don't want to just give myself a giant pat on the back over and over again, but I just think it's like, it's really awesome to spend so much of my time like researching species diversification and understanding, trying to get a better understanding of taxonomy, and then to like actually be a little bit of a part of it in a small way is kind of cool.
You certainly earned it. No question about it.
Thanks, Andy.
And I would imagine it won't be the last.
Well, we'll see. I'm pretty happy with just one. Andy, someone has a question for you. How many specimens are in that room and in that collection?
That's a really great question. I can guess. Our collections are not fully digitized yet, so we actually don't know how many specimens are here. There's 3 rooms like this, so what you see here is just one aisle. And in this room, there's about 30 aisles just like this, because I'm in the middle of a compactor unit. And we have 3 floors with similar compactor units. So there's at least 6 million specimens here. There could be as many as 10. And it's a rapidly growing collection. Each year we get up to a couple 100,000 or even 500,000 specimens as new donations.
And how many of those are like, do you think are new species to science?
I would say every collection that comes in has new species. It's just a matter of having somebody with the knowledge to identify them as such.
Wow.
These drawers behind me certainly have new species in them that we just haven't identified yet. And with the advancement of DNA technology and all types of technology related to systematics, really we're finding more and more hidden diversity, more and more cryptic species. And the other great thing is, collections like this 20 years ago had slightly more limited uses because we had the morphology and we could study the genitalia, but we couldn't get the DNA out of them then. Now, every specimen in a collection like this is a potential source for complete genomic DNA. And so the value of collections like this in this era of DNA technology is just greater than anybody would have ever imagined. So frequently I get a question, why do you have more than one of any species? Or, when do you say enough is enough? And that's a really hard call because the uses for these collections keep evolving over time. Now people are studying the phenotype of these butterflies to study climate change. And who knows what people are going to be using these specimens for 20 years from now. So really, I just see my job is make the resource available, care for it as best as possible, and time will tell really what it's all good for.
Well, yeah, and you mentioned like climate change. I think that's really interesting about a species like the Wahydra is that if it's found in such like a narrow band of elevation with such kind of like temperamental habitats, it only maybe the caterpillar maybe only eats like one kind of plant or one sort of bamboo, like you said. Like, well, you can learn a lot about like the distribution of that based off of the food availability. So like if the climate warms or something and those organisms have to go up higher in elevation, that's going to change the species richness. And like, you can't really know that when you only have one specimen of that species. And so there's just so many more questions we need to answer.
There sure are. So when you head into Ecuador to try to rediscover it?
Yeah, well, maybe like right now I'd go, but I've got some emails I probably have to respond to, but someday.
In the future. Fortunately, the place where the specimen was found, it's basically along a major highway, so it's rediscoverable. I mean, it's not like it's a super remote area. I mean,
oh, really?
Yeah, you can get in the car and go, fly into Quito and go halfway down the east slope, and you're in the habitat.
You can just hop out of the car with a butterfly net and
Well,
maybe find it?
that's basically what Harold did. He just got lucky. You know, we could try it 100 times in the same place and not see another one, but eventually another one should show up.
That leads to the next really good question that someone had, which is, what is the best way to find a new species if someone wanted to discover one?
The best way to find a new species is to spend a lot of time hanging around in a collection like this, because we're packed full of them. It's a lot less often that you just stumble across one out in the field. It happens, but it's a lot less common. I mean, we've got drawers full of undescribed species here that just need study.
Yeah, and it's not like, from what I understand, not really like a eureka moment when you're in the field, right? It's not like
that's really rare. No.
Yeah.
9 times out of 10, you're like, oh, well, that's interesting. And you don't realize it's new and special until you get back and you get the specimen prepared and you dissect the genitalia. I have had a couple of instances of groups that I've already studied real well, where in Mexico, when I've been in the field, and be like, oh, well, that's got to be new. I mean, that's really exciting, but that's really rare, because you'd need to know a lot about something that you didn't know existed ahead of time. So it doesn't happen very often.
Yeah. To become a taxonomist of a gigantic group, probably some sort of insect, and then you can name all the species.
Absolutely, yeah. If somebody, if your goal is to name a lot of new species, become an insect systematist. If you like butterflies and moths, Microlepidopter is the key, because probably over half of the species. Microlepidopter are like a fraction of the size of a Wahydra graslieae.
What?
Tiny little moths that are pinned on an itsy bitsy skinny little pin, and studying those is all about looking at the genitalia, and you spend your whole time behind a microscope. But if you like that kind of thing, there's thousands of new species to be named.
That's fun.
Yeah.
So we have a couple more questions here. Someone asked, which I think is kind of entertaining, which leg did you sacrifice for the DNA, and is it arbitrary?
Let me check. I don't remember. Actually, it was probably 2 legs. You tend to get more DNA out of 2 legs than you do 1, and it was probably arbitrary. I broke them off before I spread the specimen, because when you put the specimen in a Tupperware container to rehydrate it to spread the wings, that tends to break down the DNA a little bit. So I actually plucked off 2 legs before I spread it, and I think they were off the left side.
Good to know.
Yeah.
Important details. And then someone asked, why is there lag time between finding and describing new species? I think that's a really interesting question, because you mentioned that you kind of described this like in 2016, and now it is 2 years later.
Yeah, it's a really good question. There's a lot of reasons for a lag time. The publication process itself is kind of slow. You write your paper and you think you have it all done, and then you send it to an editor of a journal, and they send it out for peer review, and that takes who knows how much time it's going to take. It didn't take much time in this case. The paper comes back with comments, and you revise the paper based on whatever comments from the peer reviewer, and then you resubmit it, and then it goes through typesetting and sort of pre-publication, and so there's that whole aspect. But really, what is the holdup generally are technical issues related to the group of butterflies you're studying. So it goes back to type specimens. If you think you have a new species, ideally you want to look at the types of anything that's closely related to that, the type specimens of anything closely related to that. But in any given genus, one type specimen could be in Paris, another type specimen could be in London, another one could be in Berlin, and another one could be here in Florida. So it might take 2 or 3 or 4 years to get a chance to go to all these places if they're not willing to ship you the specimen, and usually they won't. If it's a unique holotype specimen, they'll say, no, you can come visit. We don't really want to stick that in the mail. It's too valuable. So the amount of time it takes to go around and study the holotype specimens at the various institutions really is the biggest sort of holdup. Photographs online are great, but they don't really usually tell you everything you need when you're doing a study like this.
Yeah, because you mentioned the importance of having to look at the genitalia. You can't probably a picture of that's not going to do it.
Well, you know, we do illustrate the genitalia in these papers, but sometimes you need to see it from a different angle than it was illustrated. So you got to go back to your little vial and pull out the bug bits and look at it from a different angle. So
that's time consuming. It's all that travel. Got to fly all around the world. Shucks.
Yeah, it's pretty rough.
So Andy, what do you do if a holotype gets destroyed? What do you have to just pick a new one?
Yeah, you make what's called a neotype, and it happens. The big 1904, 1905 fire in San Francisco. We lost a lot of types. A lot of types were lost during World War II in Europe. And so if your type is gone and you know it's gone and you're doing a study like this and you require a type as a basis for comparison, that's when you designate a neotype.
That's smart.
Yeah.
But that's, I mean, that's like worst case scenario.
That's worst case scenario. And if there's only one known specimen, you don't really have that option.
Yeah. Well, let's take one more question, and then I think we'll wrap up and let you get back to describing new species, which is very important work. Someone asked, and I think it's a good question too, is like, why is it important to discover new species?
Well, you know, we're losing biodiversity on this planet at a really rapid rate, and I'm sure we have lost a lot of skipper species before they were ever even described. And so it's hard to make conservation priorities if you don't even really know what's out there to begin with. And we really don't understand very well what their role in the environment is. Certainly they're probably pollinators, even though some skippers are actually nectar thieves. They probably pollinate something. The larvae are certainly hosts for parasites. They're, you know, food for predators. So knowing their role in an ecosystem is really important, especially if you're trying to save that ecosystem.
Yeah, I think that's probably the best case for species, describing species and asking questions about biodiversity on this planet, because the reality is we really know far less than we think we do, right? I mean, the number of known and described species is such a tiny fraction of the biodiversity that's actually on the planet. Yeah. So, you know, I think it's really interesting that, and it's something that I have to kind of fight against all of the time, is this misconception that we know everything that's ever lived and that new species discoveries are rare, because they're really not. I mean, there's, like you mentioned, there's more
and even
species.
With butterflies, you know, a lot of people think that, well, all the butterflies must certainly be known, but absolutely not. Yeah.
We don't even know all the butterflies.
Right.
Don't even. Well, Andy, I want to thank you so much for taking the time out of your day to chat with us here on the Brain Scoop.
My pleasure. Yeah.
And to have named a new species of butterfly after me. I think it's probably the coolest thing ever that's ever happened.
You more than deserve it, and we're just so happy that that you're happy.
Thanks, Andy, and thanks to everybody who tuned into the live stream. This is going to be archived on the Brain Scoop, so if you didn't catch the whole thing, you'll be able to tune in and watch it later. Well, thanks everybody. Thanks, and if you're in Florida, come check us out. Yeah, go check out the University of Florida Museum of Natural History. I haven't, but I need to, because I gotta go check out my butterfly. It'll be here waiting for you. Yay! Okay. Thanks, Andy. Thanks, Emily. Bye-bye. Bye.